Hello everyone. Today I will be presenting my model of DNA replication and explaining the process. I built a manipulable model to demonstrate the key steps, and I'll be showing you detailed drawings as well. But these drawings will be in the next part of the segment. This project has been a fantastic way to learn about this fundamental process in biology. Let's start by looking at the structure of DNA itself. As you can see in my model, DNA is a double helix like a twisted ladder. Each rung of the ladder is a base pair, and these are represented by the different colors. These rungs are actually formed from two nitrogenous bases held together by a hydrogen bond. For the sake of simplicity, the blue and the red are the two nitrogenous bases, and the clear part in the middle is the hydrogen bond that is holding them together. Each rung actually consists of purine, which could be adenine or guanine, paired with a pyrimidine, thymine, or cytosine. Adenine always pairs with thymine, and cytosine always pairs with guanine. These are held together by those hydrogen bonds we were talking about earlier, 2 between A and T, 3 between C and G. This base pairing is crucial for DNA's function. We're going to dive a little deeper into that later, but you can see they can be switched out depending on the structure. The hydrogen and nitrogen will change off of the base pair. The sides of the ladder are the sugar-phosphate backbone. This is represented by that blue line. They are made of alternating deoxyribose sugar and phosphate molecules joined by strong covalent bonds. Phosphodester bonds are the kind of bonds that join the nucleotides on the backbone. These bonds are a type of covalent bonds. The reason why they are used for this is because they provide the stability and structural integrity that the molecule needs. These actually represent the purines and pyridines. The green one is guanine, and the red one is cytosine. The pink one is adenine, and the blue one is thymine. Next is what I told you we're going to get into later. Adenine and thymine pair via 2 hydrogen bonds, and cytosine pairs with guanine via 3 hydrogen bonds. Hydrogen bonds are used because they are weak enough to allow the DNA strand to separate during replication and transcription, yet strong enough to hold the strands together under normal conditions. These black caps on the end are telomeres. They are on the end of the chromosome. They are repetitive nucleotide sequences. They actually protect the ends of the chromosomes from deterioration or fusion with the neighboring chromosomes. Therefore, you can assume they play a crucial role in maintaining the gene stability and are involved in the aging process and even cancer development. Now we are going to move into DNA replication. The helicase is the pink triangle, and it unwinds the DNA's double helix by breaking those hydrogen bonds. The next part is the topoisomerase oval. It relieves tension and prevents supercooling. It does this by cutting, swiveling, and rejoining DNA strands. The primase is the black square, and the primer is the striped one. The primase's function is to synthesize the short RNA primers complementary to the DNA template. These primers provide a starting point for the DNA polymerase. The composition of primers is just short RNA sequences, and they're necessary because DNA polymerase can only add nucleotides to an existing strand of nucleic acid. This yellow wonky-looking oval represents the DNA polymerase. DNA polymerase are basically enzymes that synthesize new DNA strands by adding nucleotides complementary to the template strand. The 3 versus 5 designation refers to the directionality of the DNA strands. DNA is synthesized in a 5 to 3 direction, meaning nucleotides are added to the 3 end, therefore considered antiparallel. The leading strand would be considered the green one, and the lagging would be considered the red ones. Take note of the direction they're moving in. The 3 and the 5 then listed are considered part of the DNA strand, so the fragments are actually moving in a 5 to 3. The red strands are also referred to as Okazaki fragments. This blue squiggle is the ligase, and it actually combines all of the Okazaki fragments, thereby creating a continuous strand. This pink circle is telomerase. It extends the telomere's repetitive nucleotide sequences at the end of the chromosomes to prevent loss of the genetic information during replication. Telomere formation protects the ends of the chromosomes from deterioration or fusion with neighboring chromosomes. Sounds familiar, doesn't it? Prokaryotic replication occurs in a circular DNA molecule with a single origin of replication. It is generally faster and involves fewer proteins. On the other hand, eukaryotic replication occurs in linear chromosomes with multiple regions of replication. It is more complex due to the involvement of histones and chromatin structure. Thank you for tuning in. My name is Gabby Mendiola, and remember, why did the DNA cross the road? To get to the replication fork.